Hi, my name is Lena Pernas. I'm an assistant professor at the David Geffen School of Medicine at UCLA in the Department of Microbiology, Immunology, and Molecular Genetics. And I'm one of the co-organizers of the upcoming Keystone Symposia, Mitochondrial Biology in Health and Disease this January in Taipei. So I think we can all agree about the idea that mitochondria are simply cellular powerhouses is now really a relic of scientific past. And we now appreciate that these organelles play a key role in just about every cellular process under the sun. And so our overarching goal for this meeting was to address the diverse roles of mitochondria, but from the lens of their bacterial origin. So how do these bacterial descendants accomplish constant and bidirectional signaling with their host cell and host organism at different scales? Why does the dysfunction of these endosymbionts manifest in such diverse disease depending on host cell type, tissue type, and age, despite that they do carry out several conserved tasks? So to this end, the topics we selected, we felt put these questions front and center. So I think our session topics highlight the evolution in how mitochondria are perceived. For example, a huge paradigm shift in our field that was pioneered by our keynote speaker, Dr. Navdeep Chandel, was the concept that mitochondria are not static powerhouses that produce just chemical energy, but that they are signaling organelles. And we're now understanding that signals derived from these bacterial descendants don't only influence their host cells, but also their host organism in different ways. So for example, by impacting developmental rates of an organism or even how we respond to infection. I think another very interesting shift in our perception is that these organelles are not only importers of proteins and metabolites and other signals for their host, but prolific exporters, and so have myriad of ways of receiving signals and communicating with their host. So electron microscopy was my gateway drug to mitochondria. These organelles exhibit remarkably unique and diverse morphologies in different tissues that are a far cry from the bacteria from which they originated. And yet we understand so little of how their diverse forms in these different tissues are tailored to their function, nor how they came to possess such unique morphology. So we highlight these two questions in our workshop. Really excited to see the abstracts that we will receive that are relevant to these workshops. So our two deadlines are October 15th for short talk. And if you would like to attend and simply submit a poster, the deadline for that is December 23rd. I hope that this meeting moves the field forward by raising new questions and approaches to studying mitochondria, by viewing many of the functions of mitochondria and the dysfunctions of mitochondria from their bacterial origin and from the lens of them being endosymbionts rather than powerhouses of our cells. Most of the keystones that I've been to have been in the US. This is the first one that I will be attending that is actually in a different country, and not only that, in Taipei. So I think we'll have a much more diverse audience than we have in the past. And I think the Keystone meeting format in general offers ample opportunity for interacting with peers at all stages, thanks to having most meals together from breakfast, lunch, and dinner, as well as coffee breaks. We also have career roundtable sessions that allow younger scientists to directly engage with more senior scientists, depending on the questions that they have for their scientific trajectory. And I think that the free time is actually really important for engendering new community building and friendships from scientists that attend the meetings, because it gives you ample time to get to know other peers in the field and peers that you might not have interacted with in previous meetings, or at least have had substantial quality time to do so. I do think that this is a unique meeting because many mitochondrial biology focused meetings don't necessarily import the clinical aspect, but we have sessions that range from the basic mitochondrial biology as well as mitochondrial disease and how it's targeted. And we also have folks from industry joining that will share developments in their clinical pipelines. So we think that in this meeting, we're also hoping to bridge basic biology and demonstrate how it can progress to translational work. So I do think that there is value for scientists from all niches, from academics to industry to clinicians. If you want a new perspective on mitochondria and to interact with an amazing, vibrant set of speakers from different points in their career in an amazing city with probably the best food market I've ever been to, come to our meeting. It takes place in January, and remember the deadlines are October 15th for abstract submission and December 23rd for poster submission.